Yeah, man. All right, now time for our keynote, Dr. Fleming. Dr. Fleming is chair of GI oncology at Moffitt Cancer Center in Tampa. I didn't know how to spell Moffitt until we invited him to this conference. Number of F's and T's, and the M's in Fleming, by the way. He's an international leader in pancreas cancer surgery and research. He was at MD Anderson, blah, blah, blah. Here's why I'm impressed by Dr. Fleming. When I picked him up last night to go to dinner, we had Grandmaster Flash and the Furious Five, and he just started moving with it. So I turned it over to
Memphis.
you. Yeah, it's good to see you, Dr. Fleming, and welcome to Piedmont.
All right. Thanks so much. I will echo all the comments about how important these types of gatherings are for exactly the issues we're talking about. This is a little different talk. What I'm going to do is try to weave in some principles around ecology and evolution to a lot of the stuff we've been talking about this morning. I think it really gives you a different way, kind of puts the problem on its head a little bit, and hopefully you'll know what I mean. Those are my, yeah. So that's Bob Gatenby. Bob is chair of our radiology department, but he also is heads our integration. It's a mathematical unit that is a mathematic oncology unit at our place. And he leads this effort and is a national leader in the concept of evolutionary biology and cancer. So on the right is there's a famous article that goes through all of the characteristics of cancer on the outside ring. The inside ring is the principles of evolution and ecology and how they fit with this. So this is an emerging way of thinking about cancer that I think we would pay attention to. You know this. This is the projected survivals on the left-hand side where we don't want to go by 2030. One thing I'll note is that the curves that are all going down are cancers that have a detection strategy that's widely used, and we don't have one yet. The problem on the right is that we're not improving survival after people get it. So those are all cancers ranked by the change in their survival rate from 1970s to 2013, and we're at the bottom. Hopefully since 2013, we're doing a little better, but we got work to do. So what about the origin of pancreas cancer? So the parallel in ecology is a selection process. So a fitness survival process. The first thing to understand, and we've talked a lot about DNA this morning, mutations are part of life. They're part of developing of species, adaptation. They're happening all the time. So this is a great article in the New England Journal that talks about this. Each of those, in that model, each of those circles is a mutation. The bigger the circle, the larger the mutation or the larger the impact. And it can be influenced by inflammation, which keep that in mind. And then you can get changes that actually help the organism adapt to that inflammation, or you can get damage and you could get a cancer. So they're overlapping activities that are happening all the time. All three of those, mutations, inflammation, cancer, are all one in a big spectrum. So we started a study where we took, so people will get individuals who are declared brain dead, their pancreas will be harvested and then transplanted to someone else. But they don't always use the organs that they harvest. So with an agreement with our organ bank locally, we started looking at these pancreases that had been harvested and not used. This is the pancreas. So this is the whole cuts. So that's an H&E of a normal pancreas from a mid-20-year-old who donor. Take note of the organization. Everything has its place. There's These acini are all intact. This is a 60-year-old. This person doesn't have pancreas cancer. He died of something else. But these changes are all abnormal, markedly. Those are all precancerous, that Pannin lesion, the ADM, all those things marked with the light blue, those are all precancerous lesions in a normal pancreas, in somebody who died of a heart attack or something else. So what's happening? So this is the model of cancer development that I think kind of sums up what we think is going on. So if you look at the, there's the normal pancreas, there's the acini that are making enzymes and doing their thing. And then there's inflammation that happens, and you get this acinar to ductal metaplastic change that then progresses. And look at the timeline at the top. So that's 20 years of development, 15 years, 17 years. This is a long process where cancer is developing. Where the problem is, we're at the very tail end when someone gets diagnosed. So we don't have much run up, and that's what we're trying to improve. This is consistent. So what we found in the donor pancreas is consistent with what's been seen in autopsies from back in the early mid-2000s. This is a Japanese study where they did autopsies on people who died of any cause, 90 autopsies, broke them down into decades, and you see those Pannin lesions occurring in 90% of autopsies of someone over 40. So this is in Japan. So if you took 1 million people and you looked at that and you looked for that, there'd be 150,000 people who had Pannin 2 lesions. So kind of the mid-development, walking around, going to the mall. So this is happening all the time. What is going on here? I want to introduce a concept of, it's called convergent evolution. So when there is pressure from the environment, the body mutations are happening all the time, right? So the classic model here is sickle cell. So sickle cell anemia is endemic to the same regions that malaria is endemic, because it's an adaptation that allows people to live in that region of the world and not die of malaria. So the pressure of having malaria. So think of the inflammation in the pancreas as like the malarial pressure. And so changes are going to happen. Mutations are going to be adapted so that these cells can live in that environment like the pancreas that I showed you. This is a principle that's known in evolutionary biology. Basically, you have a core habitat. So look at the one on the left. There's an adaptation that has to happen. If a trait happens and it's really important for survival, then that population can grow up like the sickle cell trait. If it doesn't help, it doesn't really become separate. So what does all this have to do with pancreas cancer? So if you take those early lesions that I showed in the pancreas and you take them out and you look at the DNA, they all have KRAS mutations, every one of them. It is a driver, it is a gateway, or GNAS is another. These are gateway mutations. What I mean by gateway? Well, they allow the cells to adapt to that environment to live. If they don't have it, they die, which is why you don't see them. If they have it, they live. And then if you look at that graphic on the right-hand side, if you look going through from Pannin 1 to Pannin 3 to invasive cancer, the density of the cells that have that mutation goes up markedly. So in other words, the populations are repeatedly adapting by having this mutation more and more present, because it helps the cells survive. Now, KRAS is a really interesting molecule. It's small. It's not a big gene. These are DNA structural mutations. And if you look at cancers, they're widespread. 30% of cancers have this mutation. So it's not just the pancreas, lung cancer, colon cancer, it's an adaptive change that's present. Now, the really interesting thing, if you look at the type of mutations, if you look at that graphic on the bottom left, there's really only 4 or 5 types of mutations. So that's a single base pair chain change in the DNA of that one gene. That change is present, is the dominant change that you see. In other words, it's not just any mutation, it's those in particular. So why does that happen? So if a mutation or a trait really helps animals survive, then it keeps reappearing. This is the phyla of reptiles. And each of those little stars that I made, that's a venom type that they develop. And those venom types give them the ability to adapt to their environment by killing prey or warding off somebody that's trying to eat them. And so these mutational hotspots are going to recur if they're helpful. So something about this KRAS mutation is helpful. And if you look, so you can do experiments where you take all these different KRAS mutations on that top left-hand side. Those are all different ways you could mutate the gene, but none of them are really helpful for the cells to transform into cancer except the right-hand side. So those are the ones that matter. So the point is that those mutations allow cells to live throughout that spectrum and are super important. And it's a biologic principle. It's not just random that they all have that. So this is really fascinating. So we talked about drawing blood on patients and looking for KRAS. So they did this in this experiment. So this is healthy people with chronic pancreatitis, people with pancreatic cancer, top to bottom. They drew their blood and they checked for mutations in all these different types of KRAS, mutations. And what you notice is at the top in the healthy people, they have them, first of all. So just like our autopsy study we did, there's people with mutations walking around that are in their blood. But the interesting thing is the variety of the mutations is much higher in that group than in the cancer group. The reason is that those mutations become more and more enriched because they help the cancer cells survive. So could this be a screening strategy? Not ready for prime time, but potentially if you could sequentially measure, you might be able to tell somebody, hey, your profile's changing. We need to do more investigation, maybe something like that. I want to touch on cystic lesions. So what about them? How do they relate to all this? So cystic lesions are think of them as another version of what I just showed you. They're just another expression. If you take animal models, you can make them make pannin lesions like I showed you, or cystic lesions by changing the order of the gene mutation happening. It's just a difference in sequence. So you can get cysts like this. We know that, I just wanted to touch on this, that there is now data that you can use clinically of risk of mutation or of cancer developing in a cyst over time. If it's a low-grade cyst, that risk is about, and I think we kind of quoted it, it's about 8% at 10 years. If it's a high-grade lesion, it's about 25% at 10 years. So you do need to hang on to these people because their risk is not changed. It doesn't go away at 5 years, even if they don't have cancer. So, and that's because these cells are adapting. We talked a little bit about this. This is the Pan-seq study. This is the DNA study we were talking about where you draw the cyst fluid off and you send it for mutational analysis. This is what you get back. But what we learned from this is that it's not just the KRAS mutation, but it's the KRAS mutation plus. If you start adding mutations in that P53 gene or the P16 gene or the SMAD4 gene, what's happening is you're pushing that graph to the right. You're adding capabilities to those cells. capabilities like learning how to metastasize or go somewhere else, which you don't want. So the way we use this, we sequentially sample and we'll send it for DNA analysis. And just having a KRAS mutation in your low-grade cystic lesion doesn't necessarily mean you need cancer, but if you start adding mutations over time, we're going to pay close attention to that. And that's it, because that is, you are progressing along that pathway. So the common thread here is fitness benefit and external pressure. So these mutations that pancreas cancer has, the reason it's so lethal is that it has been developing for years in the pancreas occultly, undetected, but fending itself off. And it has immense external pressure. So we need to look for these common threads of adaptation to counter these. So to summarize, these precursor lesions, like we're talking about, they're common in individuals really over the age of 40. You can see change in autopsy studies. Most of these don't become cancer, right? There's only 40,000 cancer cases a year in the United States. So you can imagine there are millions of people who have these changes who never get pancreas cancer, right? So it's not like it's happening all the time. Your body is kind of in a struggle. It's all about fitness. KRAS mutations offer that substantial fitness, and then silencing of those secondary genes is really when it takes off. That's that part where it really ramps up. What about growth, survival? So once you get a cancer, how does it grow and in the body? And because that's how people die of pancreas cancer. So to me, the great ecological parallel is invasive species. So an invasive species is a species that was maybe growing in another part of the world and was moved to a different environment and now is growing unchecked. And these are 3 examples in the South. I thought you guys would like these. I saw some kudzu on the way from the airport here, overtaking a house, right? And then that water hyacinth in Florida, a woman dumped these. She thought they were pretty, so she put them in the St. Johns River, 100 years ago, and now they fill. That's a canal in the bottom right-hand side full of this invasive species. So things that they can do: rapidly proliferate, destroy existing habitats, outcompete native species for food, no predators. They usually don't have predators. So this is a cartoon of normal epithelium, cancer developing epithelium, invasion. And what's happening is it's not just the cells, it's not just the cancer cells, they're changing their environment around them. And I'll take you back to this guy. So this is our 60-year-old. You see the little areas where there's like a round bunch of cells, but look around the outside of it. All that is like scar tissue. That's not normal. It's inducing that change in normal tissue. The cancer cells are. They're changing the environment. We work with this molecule called DDR1 in my lab. So DDR1 it binds to collagen. So cancer cells actually interact with that scar tissue that you're seeing, and then they make chemicals that call in white cells that then chew up everything around it. So they're basically modifying I call it building a house. When you see a tumor, when you see a pancreas cancer on a CT scan, most of that mass is not cancer cells. Most of that mass, 90% of it, is that stuff. Is this interaction where there's like a scar around it. That's why it's so hard to get chemotherapy things into it. So KRAS is actually the driver of this stuff too. So this same mutation that we talked about from the beginning is the reason it's so present is because it's an ultimate adapter change. So those mutations, this is a map showing you all the signals that are related. Look at that one on the right-hand side. That's like interacting with every function of the cell. So it's basically hijacking all these normal functions and making them help it survive. And how does it do that? Essentially, it's a metabolic machine. Cancer cells have to divide rapidly. So what they do is they take every piece of energy they have to make new DNA, to make new cells, because they replicate over and over. So KRAS, you see the red arrows, it's in the middle of these pathways. That image on the right-hand side, that's from our lab. That's a cancer cell essentially harvesting surrounding molecules and then chopping them up to harness for food. It's essentially like strip mining the surrounding environment to get nutrients to make more DNA. And you can do this in mice. This is a study where they basically increase the amount of that KRAS mutation, and you see those changes from left to right, from the low, mod, high. They're basically making this change happen. And so it's not just the presence or absence of the KRAS mutation, it's how many copies the cell actually has. In other words, it's like a dose effect. And we showed this here. So the left-hand side there, you can now do these experiments where you can actually measure not just the presence or absence of the mutation, but how many copies of the abnormal gene are present. So it's not just the yes, no, it's yeah, how much. And what happens when you go higher, the effect is greater. More and more and more and more. The cells actually become resistant to treatment. That molecule in the middle, that's a new drug that has been touted as it treats KRAS. It actually treats this change we're seeing. And what we're showing on the right-hand side is that if the mutant copies are super high, the cells are resistant to that drug. In other words, they can overcome because they can basically go around the drug because their copies are so high. The good answer is if the copies are low, the cells are sensitive. So we're hopeful that this will become you may read about a KRAS specific inhibitor. This might become an everyday drug that we use to treat patients. But we have to the idea here is to get smarter about who's going to benefit from it. So what's going on? So how's this what does this have to do with like invasive species? And so what's known in ecology is that the reason you have like the big animal and the little animal living next to each other is they have their own niche. They don't compete directly. When you get an invasive species, it basically competes directly with the little animal, so it pushes them out. And that's what's happening here. When the niches overlap and they're directly competing, you get that. Great example here, this is the rusty crayfish. People used to use these, or still do, use these to fish in the Midwestern United States, but they're not endemic to the Midwestern United States. And people when they would finish fishing, they would just dump them into the water because they were bait. Well, what happened is you got left-hand side at the bottom, that's like the normal bottom of a lake. You got all the fish and the fauna, everything's organized. The right hand is what happens when these crayfish, because they eat everything. They eat all the plants, and then the fish can't survive. And so you have barren lakes all in the Midwest from this. So think of that example as an example. So growth summary. Rapid proliferation of cancer cells allows them to outcompete normal cells for substrate. They're basically competing for like oxygen, glucose, all those reagents that they need to survive. They're just doing it faster. They're growing faster, they're competing faster. Cancer cells can adapt to they shape their own environment. And then that dosing of KRAS is going to be more and more and more important. We don't measure that now, but we think in the future that's going to be critical, especially when these new drugs hit the everyday market. Okay, pancreas cancer is bad. We know that, right? So what about treating it? So if these principles are true, could they be used to treat? Can we use them to treat? 99% of species on the earth have become extinct over time, right? There's no dinosaurs walking around. There used to be. There's not anymore. So these are all species that are now extinct that used to live on the earth. And so if you think about what happened to them, you can maybe use that for treatment. This is the model that we use, that is used for, so ecologists use this model for whether to understand where a species is in the evolution of extinction, in its extinction. So the concept is this: you have a population size and it's cruising along, and then there's something called a first strike, right? Habitat wrecked by something, right? Meteorite hits the earth, whatever. Some first strike that causes the population to drop rapidly down to that area where you see the dotted line. And then that's kind of a no fail line. They call it the minimal viable population, MVP. And ecologists know that if the population drops below that, they'd have a 95% chance of becoming extinct. There could be recovery, though, where the species comes back. But there's some consequences to that called genetic bottlenecking, where if a population gets really low, the gene pool in that population becomes very narrow, right? And so those mutations that are happening all the time, they actually become more pronounced because all those four animals that were left now become 40, but they're all descendant from the same four. So they're all very, very similar, and they're susceptible to death, to dying from another cause. So that's called a founder effect. So here's an example. This is the heath hen. Doesn't exist anymore. Used to be on the upper eastern coast of the United States. Some people believe this was the original Thanksgiving turkey because they couldn't fly and they were easy to catch. They were easy to hunt. So people ate them. So the population went down. So to save them, they took the last remaining heath hens that they knew about and they took them to Nantucket, to an island. They were going to preserve them. So you get the founder effect where they grow back up. But these heath hens were all descendant from like a small number. They became victim to predation, or they had a fire. So then they basically went extinct in 1925. So just as an example. So the concept here is you want that in a treatment context, you want that extinction vortices to happen. In other words, you want to get that population down low enough to where you can finish them off, the cancer, right? We're trying to make it extinct in the body. And this is called the extinction vortex. So here's the challenge. If you're looking at this is the staging of pancreas cancer. If you're looking at a T2, T3 primary tumor, that's about 100 grams of tumor. That's about a 2-centimeter tumor is about 420 million cancer cells. A 4-centimeter tumor is about 3.4 billion cancer cells. So it's like taking the population of mice in the world and trying to make them extinct. So it's a challenge, right? So how do we do that? Because like we said, we're finding this late. So how do you reverse that? The other challenge is that these cells are not static. This is just a simple thing we did where we grew cells from a patient in the lab. And what happens is we saw that as you grow these, they will change. They're not sitting still. They're losing characteristics. They're gaining characteristics. They're adapting. So when you treat, it's not just one treatment and then you stop, right? Because the cells are adapting, so you need a sequence. So when there's this recovery, there's two ways it happens with treatment. One is metastasis. So the cells leave their original environment, go to a new environment, like the Galapagos finches, traveled from South America, right, to a new environment, allowed them to live. The bottom, those are lemurs in Madagascar who used to live in an area that is now overrun with development. So now what happens is you have these little pockets. Those little gray areas are little pockets of lemurs that are trying to live within a city. On the right-hand side next to that, that's a treated pancreas cancer. Looks familiar, right? So you have these little cancer cells that are surrounded by scar. Those guys are still alive, but they're on the ropes, but they're still alive, and they could come back, right? So we need to complete the extinction. Even though we're trying to save lemurs, we're not trying to save cancer cells, right? We want to kill all those. So metastatic seeding is this is sobering statistic here. This is a study where they actually harvested portal vein blood from people who had a pancreas cancer in the pancreas, and what they found is circulating tumor cells were present within that blood, such that it was about 62 cancer cells per 7 mLs. If you do the math, so the portal vein flow is about 1 liter a minute. So if you do the math on that, that's about 8,000 cancer cells leaving the pancreas tumor and going into the liver every minute. So it's constantly shedding these cells. Now, the good news is that your immune system is really great at killing those. Like 99.9% are killed by your own immune system, so you don't get cancer in the liver, but all you need is 1 or 2 to live, right? So this is where, and this is true, this is a recurrence pattern. If you look at patients who've had curative surgery, in year 1 there, the liver is the dominant site of first recurrence, and that's why, because it's being seeded with these cancer cells. So I just show that because it's going to matter when we talk about treatment strategies. Okay, so one of the things that happens is that founder effect. Those metastatic tumors are subject to that founder effect that we talked about, because 1 or 2 cells get out and then they grow into a new tumor. Those cells are actually more sensitive than the original to therapies that you would give, which is often why you see if a patient has a pancreas cancer and has a liver metastasis and we give chemotherapy, sometimes the liver metastasis will go away, but the primary tumor hangs around. Those cells are more sensitive in the liver because of this founder effect. They're more fragile. So there's a couple different strategies of how to treat pancreas cancer that's localized out there. One is you do surgery and then you give chemotherapy afterwards. Well studied, actually some great data, and I'll show you that. How does that fit into this paradigm? So if you do that strategy, surgery is your first strike. So your first strike is to go to the operating room and take out the primary tumor that you believe to be the main source of cells. And then adjuvant therapy is going to prevent that recovery, right? You're going to add chemotherapy and it's going to prevent those cells in the liver from growing up. So the concept is surgery first gets that bulk, adjuvant therapy gets those cells that are in the liver. You drop that liver as first site of recurrence, right? So it's very important that they receive that chemotherapy on the backside. That's been a strategy that's been around for a long time and has worked for a lot of patients. These are all the studies looking at that strategy. So surgery followed by chemo. What you notice in the overall survival column is improvement, but not really getting better. We're like, yeah, yes, this strategy works, but it's not improving. Those are over time. Like that top one, the Gitzik study, that's the old one. You don't see a whole lot of movement, right? I think we've proven to ourselves that this is happening. This is a common pattern. Why might that be the case? So this is a study we did. It's not a prospective study. It's retrospective. But we took patients who had surgery followed by chemo, what we're talking about, or chemotherapy and then surgery. And the first thing is that about half of the patients who get surgery first never get chemotherapy. And that's true in most of those studies that you're seeing. The surgery is a big deal. Patients are fatigued. They don't want to get chemotherapy, so they never get it. And what happens if you get surgery and you don't get any chemo, you fall into that survival curve that says unsuccessful on it. If you get surgery and you get chemo and it goes well, you fall into that successful survival curve. So there's a big difference there whether you get chemo or you don't after surgery. Why does that happen? Well, so if you get surgery and you have a complication after surgery, your immune system is not the same. This is studies from trauma, from like people in auto accidents that has been known for many years is that if you get a series of traumatic events, so let's say you had surgery and then you got sepsis afterwards, what happens is you get a profound suppression of your immune system. So all those cells that are out in the liver, they're not getting killed anymore efficiently. And so when you hear about people who have surgery and then rapidly recur, this is often the culprit. So it's super important that you have surgeons who can get you, like these gentlemen that are here today, get you through that surgery and have a good outcome on the other side. It's very important from a cancer standpoint. So this is probably the best surgery plus chemotherapy survival data in the world. It's called a PRODIGE study. I want to point out a couple things. They use FOLFIRINOX, which is the state-of-the-art chemotherapy after surgery. Had a 54-month overall survival. Great results compared to historical that I showed you. What's different about this study? Well, they were randomized after surgery, after they had recovered. So they didn't randomize people before surgery. They randomized them after they were better. And that is a big difference. And so this shows you that, yeah, if you can get through surgery and you had a good outcome and you get chemotherapy and you can receive it all, we can have really good results. It's just a little bit of a double jeopardy that you're going to get all that, right? So what about flipping it? What about giving chemotherapy first, then doing surgery? So if you go back to our model, if you give chemotherapy first, pre-op therapy is the chemotherapy is the first strike. It goes to the primary tumor, it goes to the liver, it goes to both places. Surgery is really kind of the external event. I tell patients it's like we're taking out the trash. We're the last people. If we say that those, and this plays on the principle of that founder effect. If the cells in the liver are more sensitive and you give chemotherapy and you feel like you can quote sterilize or reduce that number down to that MVP in the liver, the primary tumor is probably going to have some cells hanging around like the lemurs, and you need surgery to get those last cells. So that's the principle. Is you're basically using pre-op chemo and surgery pre-op chemo to both, and then surgery to get those last remaining populations in the primary, not so much in the liver. So what is evidence that this strategy, how would you know if this is working or not? So that was one of the big questions we always had is, if you're gonna give chemotherapy, what is that first? How do we know that it worked? One is to look at the tumor after you take it out. So we get a report card. So I tell patients of how that chemotherapy worked, and we know what you're looking at is survival based off of response. That top curve, the better curve, Those are patients where they had less than 10% viable cancer cells in the tumor when we took it out. The other ones at the bottom, they had more. So you do get a report card of that, but it doesn't really help you while you're giving chemo. And this is where we talked about CA19-9. This is where I think it can be very useful. It's not great for screening. It's not great for random numbers, but we think it's powerful to compare your original CA19-9 to the one after you got chemo, or even while you're on chemo. So what this is on the left-hand side, that's the pre-op CA19-9s, and on the right-hand side are the same patients post-chemo 19-9s, and then they're grouped into 4 groups based off of what happened to them. Didn't get to the operating room because they progressed. Got to the operating room, but the tumor came back in 2 years. Operating room greater than 2 years, cures. The no recurrence yellow box. And what we found is that in that group, we had a very much a normalization of CA19-9 with the preoperative chemotherapy. So it's not so much that you start with a 3,000 versus a 300. It's what happens when you get chemo that matters. And we think that reflects the response that we're getting in the liver, primary tumor, everywhere. So it's a useful number. This is a randomized study, so there hasn't been a lot of comparisons of that pre-chemo or surgery up front versus pre-op chemo. This is one from the Netherlands. They randomized to receive chemotherapy and XRT or surgery up front. And they just published their longer term survival, and there's definitely an overall survival difference with getting chemotherapy up front in this study. Now, it's not massive, but it's a difference. One reason it might not be massive is if you looked at the chemotherapy they used, gemcitabine is not FOLFIRINOX, probably. In other words, so you could this implies that you could go up on the drug that you're using and you might get better results. This is a meta-analysis of other trials that have been done also supporting that approach. Okay, so chemotherapy, radiation, surgery, we've been doing those for a long time. What's new? So what do we need? So if you're an ecologist, you're thinking about it from an extinction standpoint, what killed the heath hen? Foxes, coyotes, predators. We need predators. The best predator in your body to kill cancer cells is your own immune system. T cells. T cells are the reason you didn't die of COVID and you're here, because they killed it, and because we probably all got exposed one way or another. And so this is a Nature study by a surgeon at Memorial Sloan Kettering, Dr. Balachandran. Basically, it's a vaccine trial for pancreas cancer. So they basically took the tumor. This has been, by the way, this has been done in melanoma for years, but we're now getting to where we can adapt this maybe to pancreas cancer, which is really cool. Basically, they take the tumor out at surgery. They take the RNA. They look for something. They're called neoantigens or antigens, and then they make a vaccine for those, and then they give them to the patient. So basically, they had 34 patients. They did surgery. Surgery was the first strike in this model, and then they gave them therapy afterwards, and then they got down to this MVP, and they had 15 patients, and they got immunotherapy plus vaccination. So, release the wolves, right? So when you give a vaccination, you're basically priming the immune system to identify cells that have those antigens on the surface. Super powerful. And that this is the same tech that's used for the COVID vaccinations. Exactly the same. And it's been around for melanoma. It's the same concepts. These are the results. So they had 8 responders and 7 non-responders. So when we think about it, one way to think about this stuff is not, and I'm learning about it, it's not just one antigen. It's not like there's like the home run antigen that if you find that one and you give the vaccine for it, all the good stuff happens. It's probably a pool of hundreds of antigens. Antigens are just proteins that are presented to that the body recognizes as a pathogen, essentially, right? But they're all folded around, they can be shaped different, they can have different presentations. So this is kind of this was a phase 1, by the way. This was just for safety. They weren't even looking for efficacy, right? So they're just getting started. But look at that. So if you had a response, that's a curative response. And that's the difference between immune therapy and the other therapies that I was talking about. Immune therapy, you always get that tail at the end where it doesn't come back, where you cure people. If we have people here who've been cured, who are pancreas cancer curers. It is not the therapy that you received pushed it down to that MVP, but what cured was the immune response. That's the durable cure that we want. So I'm very excited about this. I think this is, so what I'm telling my patients, just stay in the game, stay in the discussion, keep yourself fit, keep yourself ready to go, because this stuff's going to be ready sooner than we think. And and I think this is probably how we're going to use it, is you're going to get all of your therapy MVP down here, and then you release the wolves. Then you give them that vaccine, and but they need to be fit enough to have an immune system. We can't give them, as surgeons, patients who aren't fit enough to be able to do this. They have to be, the patients have to be in good enough shape to be able to do this. But so treatment, I think you have to be careful about incomplete therapy is a big thing that I see in pancreas cancer that is keeping patients from reaping the benefits. What I mean by that is therapy where we've caused more toxicity than we want, and so then they can't receive what's next. And so now we've shortchanged because we didn't sequence. The other is we don't stage correctly. So we start therapy and then we have to turn it around. That's very difficult to do. Most of those patients don't do well. And so incomplete, we need to be careful about that. I think there's some merit to maybe giving systemic therapy up front. The data seems to point that direction. And we need to understand the subsets that we're treating. I know we're going to talk about oligometastatic disease. I think that's a case where those patients, metastatic patients, are not all equal, and we need to embrace that and understand it. And then I think the immune response is that's the next frontier for us. And these are all the people that did all the cool work. Great partners, great people, and we're hoping to help more patients. Thanks.
Thanks, Dr. Fleming.
You doing questions or no?
Oh yeah, I'm sure quite questions. Dr. You want to start with Dr. Szabo? Yeah.
What's the possibility that, because when I think about this, we've done a better job at subdividing tumors in non-small cell lung cancer.
Mm-hmm.
We've done a pretty good job in breast cancer. We've done an awful job in pancreatic cancer because it's just pancreatic carcinoma ductal type. And there was I think there was a Nature article that tried to subdivide pancreatic cancers, I think, into 6 different
Yep. Eric Collison stuff.
Yeah. And so I guess the question is it possible? I mean, I agree with what you're saying, but maybe we really should also try to work on subdividing our pancreatic cancers. And fortunately, we have survivors here. Is it all their treatment that we gave them? Is it their immune system? Or is it that maybe they never even needed to have their tumor taken out in the first place?
Yeah.
And I guess that's my question. Is that something you're looking at or other people are looking at?
Well, I think you're well aware of this, I'm sure. The biggest subset is the DNA repair subset right now. That's probably the one that offers the tractable. What we're talking about is so that those genetic inherited genes that we were talking about, all of those are DNA repair genes. Those are all involved in replication repair. And so you can imagine like a cancer cell replicates all over and over and over again. And then when you treat the chemotherapies and therapies that we're talking about are DNA damaging. And so you're stressing the cells. What we've learned is that if you have that inherited, even if you have a family history that puts you in that familial risk group, those tumors the tumor that you grow in that patient, pancreas cancer, is more sensitive to certain drugs, the platinum drugs, the DNA intercalating drugs. And so that's a subset that actually gets that profound response that we're looking for. Probably what's happening is you get a great response and you kill a bunch of cells and you present all those antigens out to the immune system, just like a vaccination. You're essentially auto-vaccinating that patient, and they get this profound and durable response. The question is, are we doing a good enough job identifying those people and marking them out? You know, we think it's about 10% of patients, maybe a little higher, but we could be missing 5%, or, you know, that we're not either we're not testing, or we're not taking a close enough history, or we're just not thinking about it, and we're missing patients who could benefit from a certain strategy. I think but you're exactly right. Other than that group, we don't have some HER2 negative, HER2 positive pancreas cancer, you know. The problem is those genes that I showed you, that's it. There's like 4 or 5 genes that are mutated, and that's all you need to grow a pancreas cancer. And so that's not good enough because they're not that helpful. They don't split it enough to where we can make therapies that are specific for those different subsets yet.
We've got time. Yes, sir. Go ahead.
Especially the part where where you were discussing sort of along those lines, the adaptation profiles and trending those as being a potential source of sort of a genomic screen or marker for cancer development. I was wondering, kind of going off of that, have there been any investigations as to specific adaptation profiles that exist for tumors that are more prevalent in different locations? So head of the pancreas versus body tail, since I know the idea of a early, if not even prophylactic therapy would be kind of, hindering on that, especially from a surgical standpoint.
Yeah.
I don't know if you've seen any of that or if there have been any papers investigating that.
There's a few things out there, but nothing that I would say is kind of prime time ready. I know what you're driving at, and I think in practice, we stylize this approach based off of, at least I know I do, based off of the magnitude of the operation and what we're asking the patient to do, kind of where we insert that. But there's nothing that I see that is usable information yet. But I do think, I think the like the cyst, the cyst strategy, I think is a very good one. I think the cumulative mutation approach, because those are so prone to overtreatment. I think we have to become more disciplined in how we treat those patients.
So my name is Elisa Schrader. I'm a PhD student working on lipid nanoparticles and RNA delivery. Specifically, I look a lot at like trying to get nanoparticles to the pancreas.
Yeah.
So with that last study that you were talking about, I mean, my thought was like, it's great, except for the fact that the patients had to be like, it had to be resectable to be able
That's
to
right.
get the treatment,
Right.
right? So what are your thoughts on like the idea of actually kind of exploiting a little bit the idea of the lipid or like the lipid nanoparticles and like being able to use them as vehicles either to like carry the chemotherapy and maybe make it a little bit less harming for the patient, or using it as like a combination therapy that could potentially like, I guess, boost the immune system while the patient is receiving other like treatments that are tough on them, like chemotherapy.
Right. So I actually have a patent with another investigator to do that. The idea is that, so it's, we have a hedgehog inhibitor. So to change that environment. So the concept is that you need to reverse the environmental change, all the rusty crayfish part, right? You need to reverse that and get normal cells closer to the cancer cells, and then also treat the cancer cells. I mean, the problem with the tumor like that, the one we're showing where it has all the scar, you can give IV to IV, you can give CAR Ts, you can give whatever you want, whatever cell you want. Those T cells won't live, they'll live about a minute inside that tumor because there's no reagents. It's the bottom, it's the crayfish. It's the bottom of a lake that's barren. There's no oxygen, there's no glucose. The cancer cells have figured out how to live in that, but no other cells can live in it. So you can't do cell therapy into one of these tumors unless you reverse the surrounding area. So that's one reason they're so resistant.
Dr. Fleming, really great talk.
Yeah.
And I love the analogy that you present. And ecology was my favorite class in college. And I apologize for this question, but if you were
You
to,
probably know more ecology than I do.
you're struck, you're a clearly incredibly intelligent individual. So I want your opinion. If you're struck by, if you could just go back and find the person and stop them from dropping that hyacinth flower into that creek bed, if you take that principle, there's a lot of money in the treatment of cancer, and there's very, very little money in the prevention of cancer. So if you had X amount of dollars, where would you focus it to prevent the hyacinth flower from ever getting into that riverbed?
Yeah. So I think we've seen, first of all, you can't risk stratify 400 million people, right? So if you talk practically we have to have patients into groups. And so I think the genetics is the key to the family history and the genetic background. We need to be much more aggressive with that, but start with that. They're like the iceberg that you can go down from. So that's why we're doing that clinic to get those patients they're not really patients, individuals together to gain knowledge on the AI question, to gain knowledge on the blood biomarker question, to gain knowledge on the, we're basically sampling everything, right? And then build, I think you can build like nomograms of risk that then you can do population, then you can do the bigger studies. But until you have that, you need a high enough risk population to be able to do it first. And then you can expand, I think. The other is, could you with imaging get a risk stratification of the gland? like your basically like heart health, pancreas health. Like what's your pancreas health score? Right? And you could tell somebody, look, we're seeing changes. You don't have pancreas cancer, but we're seeing changes that put you in a higher risk group. We're going to modify your diet, right? No more Taco Bell. You know, you're going to have to change. You're going to have to, no more alcohol, 0. That kind of thing, where you're doing real interventions that could turn somebody's risk. It's a lot of work, you know, but you got to start. We got to get data. You know, we got to get basic data. So.
Fantastic discussion. You might have heard me. I'm 9 months. Dr. Page operated distal. I'm a very lucky man. I'm prepared for anything. I was a social drinker, and my GI guy who steered me to Dr. Page and others, I have stopped drinking, but I yearn for a glass of wine, and I do cheat. I have a 1 or 2 incher once in a while and a non-alcoholic beer. You did mention alcohol, which propped me up on that last 2 minutes. Can you be honest with me and tell me my future strategy on drinking, not drinking?
I mean, it's a direct toxin to the pancreas. We know that. And, you know, we're seeing 37-year-olds with pancreas cancer now. I mean, there's definitely a shift. I don't know if it's alcohol or diet or both, but, you know, I think alcohol is one of the easier ones to say, let's carve that out. Let's cut that back. When I talk to patients in like this high risk clinic and they're like, what can I do? Right. I tell them, you know, here's the no fun program. You know, if you want to do this, these are the things that we think would reduce your risk. We could be wrong. But it makes a lot of sense. Seems logical, you know?
Thank you.
All right, this is our last talk before everybody gives Dr. Fleming a round of applause. I just wanted to